sponsored by Ice Miller, represented by Steve Smith and his associates, and WCBE 90.5 FM, represented by Dan Michalko, right over here. Please thank them for their support. There's a biography of each speaker in your forum flyer today, so I'll provide just brief introductions. Our first speaker is an inventor and designer of products, systems, and technologies, and holds over 75 worldwide patents. He is CEO and founder of Inventor and Algaeventure Systems, Inc. Please welcome Ross Youngs. Our next speaker has spent nearly 30 years in healthcare-related industries, including Arthur Andersen and Cardinal Health. Last fall, he became president and CEO of Biosortia Pharmaceuticals. Please welcome Kurt Dieck. And moderating the panel is the general manager of CB 90.5 FM and host of the Amazing Science Emporium, which is amazing that he gets it done every day, Dan Michalko. On behalf of Ice Miller, WCBE, the Columbus Metropolitan Club, we welcome all of our panelists here today, and Dan, the microphone is yours.
I'm not really sure where to start with this topic. I mean, yeah, look at you, look at us. We braved Columbus's what's turned out to be the biggest snowstorm of the year to come here today and talk about algae. Algae. The bane of every aquarium owner's existence. And here we are. What could possibly motivate us to sit here and learn about this thing that only makes headlines when it blooms and keeps swimmers away from ocean vacation resorts? I mean, you know, seriously, never mind Jaws. Oh, there's algae out there. Don't go. Algae, scientifically, it's defined as a varied group of 6 phyla of primitive eukaryotic chlorophyll-containing organisms, ranging in size from the microscopic single-celled forms to the multicellular seaweeds 30 meters or more in length, characterized by an absence of true roots, leaves, and stems. I'm stimulated. Now, to the layperson, that should read, algae. It's slimy. It's stinky. It's just plain gross. And it just might save your life. Primitive cultures long ago learned that certain plants and fungi can treat disease, such as willow bark, which contains the natural precursor to aspirin. Science began isolating these natural pharmaceuticals over a century ago. Penicillin is a great example. It was discovered by Sir Alexander Fleming when he accidentally went on vacation, leaving a huge pile of dirty Petri dishes left to be washed. And when he came back, lo and behold, the mold that was growing there was preventing bacteria from growing. Incidentally, I've tried this as an excuse with my wife not to do the dishes at night, and it doesn't fly. From bark and dirty dishes, we move to the sea. The National Oceanic and Atmospheric Administration, the folks who brought you today's fine weather forecast, their Technology Partnerships Office states that, and I got to get this right, success will result in the commercial development of new and unique chemical compounds from the sea, which have benefits to human health, either through disease prevention or new treatments for disease. In other words, it's a new pharmacy factory, and that is the algae we're here to talk about today. So, let's clear the water, so to speak. Ross, Kurt, I want you to introduce yourselves with these questions. First, Ross, well, who are you? And what led you to create your company in the first place? And then Kurt, who are you, and what motivated you to join Ross in this great pursuit? So, Ross, go ahead and start.
Well, terrific. I appreciate that, and I would like to thank the Columbus Metropolitan Club also for having us present today. But what I will say is I've been on quite a path over the last 25 years, and it certainly started with plastics, and though it may seem a little bit disfocused, it lines up pretty well with where we started and where we're at today. We're still involved in plastics at a company called Inventor, which I did found in 1988, originally to make packaging for compact disc. Ultimately, we got involved in other plastic products. Then we got involved in bio-based products. And it was that involvement in bio-based products that really brought on the question, if you're going to make a lot of products in this world out of bio-based materials, where are you going to get all the biomass that isn't competing with food? And that is essentially where we started our research into algae. And what attracted me to algae originally was the fact that it was this area that was really unexplored. Technically, I think there's a book out there where they call it blue ocean strategy. When you're talking about a field that had very little investment but massive opportunity. And what we determined pretty rapidly is the massive opportunity was directly related to the fact that you get so much more productivity out of algae than virtually any terrestrial-based plants. So it can produce that biomass. And when we started to look at that amount of biomass that you can create, we really recognized that it wasn't just a product. It was virtually every product that you could see from the terrestrial agriculture. So everything from energy to materials to plastics to foods to feeds to fertilizers, nutraceuticals, and pharmaceuticals. So it is this new opportunity and this new revolution. And as we started that research, we ended up with creating some technologies. And it's those technologies that bring us today to talk about drug discovery.
Great, thanks, Ross. Now, did everybody understand that? There'll be a quiz later. So why did I join Ross and the team is that I retired from Cardinal in October of 2011. I had two great careers, one at Arthur Andersen and then at Cardinal Health. And I was really looking for to stay in the healthcare area, but to do one more big thing that was actually closer to chronic disease than where I had played in the past. And so how I looked at it was relatively straightforward. I said, okay, is it a big industry? $500+ billion in the pharmaceutical industry. Is there a willingness to pay for results? Spend $100 billion a year in R&D. Is there a problem to solve? Driest pipeline in 10 years and no blockbusters in the last 5. And you have products like Lipitor, an $18 billion product that went off patent in 2011. And is there uniqueness in how to actually attack the issue? And with Ross's technology, it absolutely has all of those factors. But also, probably more for me, you know, having a brother-in-law that died of leukemia, my mom passed a year ago with Alzheimer's, wouldn't it be great if 15 years from now, as products that we identify get developed by Pfizer and BMS and GSK and Lilly actually can make a difference? And so that's also, from a personal perspective, some of the key reasons why I joined.
That's actually, I like that. No, the reason I do is that, you know, with my work, part of my whole point is to point out how science is really about people. It's not about the numbers, it's not about the theories, it's people. And you just gave a great example of what is ultimately motivating you to work on this side. Ross, do you have a similar, what's the people side of you being involved in this?
You know, I do. Part of it is that I believe that the development of sustainable technologies, sustainable products, can be leveraged by the development of high-value products. So it always starts, any technology, any development, you want to start with the highest value possible to pay for its advancement. You don't start at the lowest value, you start where there's the most opportunity. And with this, this becomes a natural evolution of development. Could it lead to great pharmaceutical products? Absolutely. Could it lead to other products that are of a lower value into the future that can have an impact on civilization? And the answer to that is absolutely. These are technologies that will develop over time, and it'll have an impact that ultimately will be profound on this planet. You may see, and I would love to say we're going to see it, but at least we will have ancestors that see it. You may see clear water again running through Columbus because of technologies that ultimately evolve from where we're at today and allow that to occur. And those are sustainable technologies directly related to cleaning up waste, whether they're atmospheric waste or they're pollution and other waste from ever reaching the water in the first place. So you've got to start somewhere, and there is no doubt that we're starting with our technology suite in an area that has the highest value.
Cool. That's huge big picture. So let's go all the way to the opposite extreme on what is going to make that happen, and that's microalgae. So on the geek side of things, so we know how the world is going to change, what is a microalgae consortia?
Well, terrific. I'll jump right into that. And that is essentially when you go to any water that is around this environment, you will find potentially more than 5,000 species in essentially a liter of water. Now, that may seem a little bit scary, but if we went down to the river or we went to the closest pond, you would find 5,000 microorganisms or more in a liter of water. And that's individual species. Okay? Now, the reality of that is you're talking about less than a gram of biomass. And you're talking about science can't even tell you every organism that's in there, because science itself can only culture 1% of those organisms in that liter of water. That leaves 99% of those organisms that have never been studied potentially for drugs. And it's the technologies that we created that was funded by the Department of Energy that allows us to take enough of that microorganism consortia, which includes bacteria, it includes viruses, it includes molds and fungus, and of course algae, which is the primary producer. And we can grab all of that and study all of that. And then the one other very amazing thing about being able to study that entire consortia of organisms is you have the capability to see activities because of the competitive pressures. So you're not growing it in a petri dish as a single organism. It is growing in the natural environment and competing. And it's those activities that produce the chemicals that give organisms an advantage and thus can lead to drugs.
Well, and that brings up now Kurt's involvement, because you mentioned new technologies to be able to start studying these microorganisms. And Kurt, you had mentioned that that's part of why you got involved, because he's got something different going on here. So between the two of you, what are you doing differently? What is the technology that you're able to do these assays?
Yeah, it's a couple things. First of all, it's that inside of that consortia, it's what's called secondary metabolites. Don't want to get into a lot of science with you, but the secondary metabolites that are created that no one has ever been able to look at before. Why? And certainly not for drug discovery. Why not? Because you couldn't get enough of it. And so if you think about it, if you go and garner 10 or 20 pounds of dry weight biomass, and by the time you do what's called fractioning to get it to actually study, you're talking about this much. And you won't be able to do any level of pharmaceutical studies with that amount of biomass. Our technology allows you to get over a ton of it, dry weight. So now, for the first time, science can actually look at the material and have enough of it to actually do testing. Examples would be we have 13 of our compounds that we've identified. We put into Eli Lilly's, what they call their open innovation program. And we were hoping that of those 13, since this was our first shot at it, maybe one or two they'd want to look at. Well, they told us at the end of December they want to look at all 13. So we know that what we're doing and what we're finding is of interest to pharma. There is no question about that. And we have people like Dr. Peter Moeller, who's at NOAA, who participates and oversees our science lab down there. And when he first saw the biomass, and he's been studying this type of thing for 30 years, he goes, I have never seen so much bioactive activity in my 30-year career. You guys got something special here.
Now, without breaking patent or proprietary secrets, can you give a general description of what it is that's different? How are you generating that much biomass? Because from that much to a
Inventor
ton.
answer that, the one that holds the patent.
Okay.
Because those, I don't hold any of them, just to be clear. No.
It is actually pretty interesting, even to me, because what we ended up discovering is that you can use nature in the way nature has designed movements of water through systems, and you can do so very efficiently and separate out very small solids. And when you think about these organisms in the water, you're talking about things that are under 10 microns. You're thousands and thousands of inches in size. You're so small, it goes through normal filtration. Well, what we recognized and realized that water has properties, and you can move water because of the adhesion of water, and then you can separate solids. And it was that technology, our approach at looking at something counterintuitively, that we came up with patentable technologies that ultimately the DOE helped fund. Now, the original goal was to get it to where we could move 10 tons potentially in an hour. The reality of it is we can move kilogram quantities in an hour. Not enough for things like plastics or something like biofuels, but easily enough for the drug discovery that Kurt's talking about. And that technology itself is something that we are continuing to pursue and continuing to advance, but it's already at the scale and capability to already do what our major focus is. We don't have to invent anything. All we have to do is execute.
You've mentioned several times, both of you actually, that Big Pharma is watching you. Why? What is the reason they see so much potential in the biomass? Why do they think this is the way to go instead of going to the lab and synthesizing, coming up with entirely new chemicals?
Yeah, let me grab that one. Probably about 10 years ago, most of Big Pharma actually disbanded a lot of their natural products labs, and thinking that the way of the future was to take this and combine it together, and somehow it's going to kill the next cancer cell that's sitting in the petri dish. Well, history has shown that hasn't worked. And so, now they're looking for companies like ours to partner with that can bring unique compounds to them, which, by the way, we pre-screen every one of our compounds that's in our library against multiple cancer assays. So we already know before we even bring them to a pharma player that it already has been active against multiple types of cancer and antibiotics and anti-inflammatory type of assays. And so, we already know that's going to be of interest. But again, it goes back to what I said before. This has been the driest ever pipeline in history of pharmaceuticals. When you have somebody like Pfizer that loses $18 billion off of their top line from a drug going generic, very difficult to replace that. And what they're seeing is that a lot of pharma companies, to combat that, were buying compounds that were in phase 2 and phase 3 of clinical trials, and then half of those never made it through the process, and they weren't focusing on drug discovery, the front end of the process, which is where we play. And so they're all now realizing the mistakes and needing to reinvest and looking for companies that have things unique. And we're looking at a place where no one's been able to look before. We're not looking at tree bark. We aren't looking at plants. We're looking at an area where it has to naturally, similar to human cells, has to naturally fight itself, or its competitors, to stay alive. And so there's that natural activity that's going on versus if I'm pulling beetle juice out of a beetle that actually, believe it or not, is used for warts, it's not having to fight anything. And so that's that new science that's being created.
Wow, you mentioned cancer, anti-inflammatories. More specifically, what are you seeing hints of as you're doing these assays?
Well,
what are you seeing hints of that you're going to be able to, I don't know, cure, treat?
Yeah. No, we've sent 15 compounds into NCI, National Cancer Institute. And again, hoping that maybe 10 or 20% they'd want to look at. Well, to date, more than 85% of all the compounds that we've sent in, they're interested in looking at. And they have 60 screens, 60 cancer assays across 9 different types of cancer that they're testing for. Now, we haven't had one make it all the way through yet, and they're looking for higher potency. They like the toxicity, because the main reason why, if you have loved ones, I'm sure, in this room that have had cancer, is that the reason why it has to be administered in a physician's office is because of the toxicity levels. Well, algae in that consortia naturally have toxicities to it. That's why, again, Peter Moeller out of NOAA, that's his specialty, is toxins. And so between having Dr. Guy Carter, who's our chief science officer, who's retired head of natural products out of Wyeth before they were acquired by Pfizer, teamed with the best guy on toxins, I couldn't ask for a better team. And so I have 2 of the best guys that are going to be able to identify those types of compounds that are most potent and toxic for that type of research.
I'm going to jump in here and just say, too, that when you talk about the range of therapeutic areas that we can focus, when you look at a microorganism consortia, all the activity for survival, all the activity that's needed to compete is actually within a lot of disease states. So virtually the disease you can think of, we very well may have a potential cure already within our grasp. And that is really important, and that's one of the reasons that we've started to really reach out and collaborate with a number of players in the industry. And just recently, we collaborated with Ohio State and Dr. Kinghorn specifically on anti-cancer compounds for colon cancer. And that's something that has just started, but we're already happy with what we're seeing.
Wow. Can I pile on that just for a second?
Yeah.
And one other thing to be thinking about as you think about this space is that if I'm a pharma manufacturer, historically, I know the types of disease states I want to focus on, whether it's cancer, diabetes, Alzheimer's, whatever it may be. And so we will really be a utility to the pharma industry because our compounds will likely be effective against multiple disease states. And so, you know, we won't be partnering with just one of the pharma players. We will be partnering with multiple players because our compounds can be active against multiple disease states.
And that's not meaning that a single compound will hit these different diseases. It means we've got such a rich source, it leads to that factory that we talk about, that we've got a drug discovery factory, and we're going to have compound after compound that does this.
So it's not too much of a stretch, although it is speculation, to say that conceivably microconsortia could be the panacea for all ills. You know, finally, the patent medicine operative has come, and it's a good for what ails you.
I think that would be a vision that we would love to be able to grab. I think we've got some very good scientific minds that keep us well grounded in the reality, very focused, and I think that we're going to learn more virtually day after day. Do we have hope and promise? Absolutely. But you really have to achieve in order to achieve.
Well, let's get back to the grounding, to the practical side. On the consumer side of things, it's no secret that a lot of drugs, despite some of the programs that Meyer and Giant Eagle and stuff have for freebies, but that a lot of drugs are really incredibly expensive. For the consumer, could this be an answer to that? Could this help bring down the cost of drugs, both from expenses on research and expenses of manufacturing?
Let me hit on that one. Is that today it costs over $2 billion to bring a product, a single compound, all the way through the new drug approval process. And that's targeted, estimated to be $3.5 billion by 2015. This chart here that's in your materials, that shows you the difference between drug discovery and drug development. And in there it talks about the dollars associated with that nearly $2 billion I mentioned. What we will be able to do at the front end that isn't even on that chart is roughly 2 to 2.5 years of target identification that gets done inside of each one of the pharma players. Because again, They're trying to do mixtures of things to test what they're interested in. So we believe with our technology that we will literally be shortening the time frame of that discovery process from 5.5 years or 7.5 years on average to roughly 4. And so that will, on each drug itself, that will pull somewhere between $250 million to $350 million out of the cost. Now, the real question is, will that drug company pass that through to the benefit of the patient, or will they pass it through to the benefit of their shareholder? That's the question we do not know the answer to.
Well, let's get that to your side. Obviously, you will need investors or have had investors to help launch things. What do your investors say when they look at what's a typical conversation with an investor? You've got a new technology, so it's not like you can go through the standard market and just look at the price to earnings ratio or something like that. What do you tell investors? What is attractive to someone to invest in this?
And it really is some of the comments I mentioned earlier. Is it a big market? Yes, $500 billion. Is it somebody that's willing to pay? The average license, and again, I think there's, here, but the big one's back there, but on average, when pharma goes to license a compound, if one makes it all the way through, which again, only 5% do. So of the 20, as hypothetically, of the 20 that start in, only one of those is going to actually make it out the other end. But if it does, as the licensor, you get a little over $400 million of earnings without really doing much. And because you're the one that created it, you own the IP, and once you license it, you don't have any further obligations. But also, if you then do the waterfall and say, of those 20, 3 of them are going to die at the lead stage. 3 more won't make it past optimized lead, preclinical, phase 1, phase 2, phase 3. If you do all that math, on average, each compound is worth roughly $20 million. And so, there's huge economics in here. Why? Because you're solving very big problems. And so it really comes down to big market, willingness to pay, and an economic model that is very efficient on our behalf, and we're rewarded accordingly.
One of the things I'm going to jump in there too is when you look at social investing, and there are a number of people that will actually have a strategy to socially invest, you may not know exactly where and how those funds get put to use. The one thing about in choosing companies that are doing specific things, you certainly can keep your thumb on them in a way that you may not be able to do if you're, let's say, investing in other ways. It's great to be able to connect a opportunity to serve mankind in some way, same as you invest your dollars. And I think all of us philanthropically do a number of those kinds of things. The real question is, can you also do it in a manner where you can keep score? And this is a good manner to keep score when you look at the kind of things that companies like ours would be doing.
Yeah, I had a gentleman mention to me just 2 days ago. He goes, I would rather invest in this business, not only because I'm going to get a great return on my money, but because if I go and contribute $1 million into cancer research of a nonprofit, 1, I don't know if it's being used efficiently, and 2, I get no real reporting on how my money is actually making a difference. They will be, our investors, we're looking for truly passionate investors that really have a reason to want to be part of our group.
So in a way, even the investors are mission-oriented. I just wanted to pause really quick. We've only got time for a couple more questions because we're about 5 minutes away from the Q&A portion where you can ask some cool questions. So stand by and be ready for that. First, because time is limited to that, for those who want to learn more after the forum, what's the best way to find out more about what you're doing?
We have a couple social media outlets out there. We have biosortia.com. We can be contacted that way. We also have a Twitter account. So we post our news as it occurs onto our Twitter account. And I will tell you that we're going to have probably 3 more announcements in the next 3 weeks. So we did just announce the Ohio State announcement last week, and there was a little bit of press release on that. But we understand that engaging people to understand what we're doing is an importance. So we can be reached out to, and essentially we will communicate with you directly as well.
My final question is more dreamy, because it is the last one. And I really want to, and I want both of your inputs on this because you come from different perspective. Say it's 3-5 years from now, and CMC asks us back to do this exact same panel. What is going to be different? What will have happened, without revealing the announcements coming in 3 days? What do you foresee is happening within the next few years in your field? What will we be talking about when you're sitting up here again?
As it relates to Biosortia, my view would be is that we will have licensed probably 20 to 30 compounds, and we will know exactly what disease states those are in, how many are impacting cancer, and hopefully by that time frame, actually many of them will be in the preclinical stages. And so we will be already getting a glimpse into the future around which disease and chronic diseases we're going to be able to impact.
Excellent. I'm looking forward to that. Ross?
I'm going to go a different tack from the drug discovery, but the reality of it is the company itself, Algaeventure Biosortia, has its technologies. And right now we have two very large multi-billion-dollar companies that are looking at our technologies for all those applications south of pharmaceuticals, nutraceuticals, foods and feeds, things like that. So we have our technology and our equipment that's patented deployed in areas around the world right now. And I think what will happen is in that 3 or 4 year period, if one of those companies invests an appropriate amount of development to get it out of that valley of death, it may very well be starting to be used to pull biomass out of polluted lakes, to pull biomass out of growth systems and turning those into products. So visionary-wise, we see a very valuable combination of what we've been able to achieve to date. Certainly 3 years from now, we'll be able to talk all about it.
That's fabulous. Thank you. And I got to say, for me, it's very refreshing to talk to two people who are mission-oriented. It's so rare. So many people focus on how much money you're going to make, how much notoriety you'll receive. But I love the fact that you've got a much bigger picture than just what's happening in the next quarter. So thanks.
Thank you.
Thank you.
It is time for the Q&A section. As promised, we'll take questions from the audience now. CMC does record the forums for statewide broadcast on the Ohio channel, so don't say anything you don't want your parents to hear. Forums are also available on YouTube through CMC's website. While you're at the microphone, please start by introducing yourself so we know who you are, and we thank you in advance for not making long editorial comments. So let's take our first question.
Were you teeing that up because you know him that well?
No. I've never seen this man before in my life.
Thank you. I'm Jim Rutledge from the law firm of Bricker and Eckler. I appreciate your sharing your vision of the future. I was curious, what do you see as your biggest obstacles to achieving the bright, beautiful future you envision?
It really comes down to throughput. I mean, it is as simple as that, is having the capabilities, the capital to actually end up doing multiple harvests a year, so that we are literally generating 1,000 compounds a year, of which 200 of those would end up having pharmaceutical interest, and then typically about 5 to 10% of those would become licensable. And so it's all about throughput, and we have the people, the technology, and the capabilities to do that. And so that's why we're, quite honestly, in a second race.
So as I understand what you're saying, the throughput obstacle is really a capital formation, capital investment.
Yep. No, as it relates to the ability to get, literally tons of biomass, not an issue. The ability to identify different places to go harvest because we have such a great team in partnership with NOAA. They know all the right lakes and streams and parts of the ocean to participate in. And so, because every place we go, we will find different types of compounds.
So in a perfect world, how would, if you could write your own script, how would you generate enough capital to generate the throughput to accomplish your mission? Well,
the great thing is that it doesn't take that much capital. We have such a efficient process because of the technology, is that, we don't need $20 million. We don't need $200 million to actually drive that throughput.
Thank you.
I'm going to add one more thing to that, too, is when you look at the market itself, there is one mid-level, smaller pharmaceutical company that goes on 7 seafaring voyages a year, spends about $200 million to go all areas of the globe to grab samples of microorganisms to see what they can grow. And again, they're only going to grow 1% of what they actually come in contact with. So when you talk about efficiency, we can go down to the riverbank and find the kind of compounds that the medical industry and the chemical industry, so agrochemical industry and also industrial chemical industry, has never seen as compounds that have activity and potency. We are in an incredible advantage. That's why it doesn't take much capital.
Thank you.
Thank you.
I'm Ken Thompson. I'm an architect. And being so, I'm thinking, like, what's this company look like? I envision these pond scum suckers that are out there, doing this sort of thing, and you've got a warehouse or something up in Dublin with, big masses of crap.
Those are actually the terms we use too.
What's this thing look like?
Go ahead.
Well, it looks 2 different ways, because when you talk about the technology side, that's one look. When you talk about the business side, that's another look. And what I'll give you a little picture of is the technological side. We can go anywhere on this globe efficiently and explore the microorganism consortia efficiently in a manner no one has ever been able to achieve. So in 7 days of Grand Lake St. Mary recovery, we were able to get more than a ton of biomass. And this is microorganisms. This is the smallest kind of filter that you could come up with. It'll pass right through. So we're in a position where we're going after those single cells that are in this environment that nobody else has ever been able to pull together and do studies on. And if we got challenged to go do this 20,000 feet under the sea, we already have the technology to do it. It's a matter of setting it up and implementing it. So there is one warehouse in town that holds multiple skids of frozen, preserved biomass right now. And that biomass is what we send to be processed to reduce it through extractions and fractionization down to those compounds that have never been seen before.
Hi, I'm Peter McRae. I have my own national architectural firm, McRae Architecture. Ross, you touched on my question. Are we going to be able to save Grand Lake St. Mary?
There's a combination of things to do that. We've looked at that and said it's an opportunity. It produces about 500 tons of biomass a day. So it has the capacity and the capability to be quite a little producer. Now, put that in perspective, the western basin of Lake Erie, which has the same productive capability and is producing at that same rate, is more than 30 times the size of Grand Lake, and Grand Lake isn't that small. So the reality of it is a lot of our nutrients, whether they're coming straight from the air because of all the fossil fuels we're putting in there, are making the way to the waters. And it's algae that's cleaning it up. So we are at the beginning stages of coming up with the technologies that allow us to have clean water in the future.
Hi, thank you guys so much for coming. My name is Lauren Eider. I work with Battelle. I'm a chemist by training. I actually was involved in natural products drug discovery as an undergraduate, looking at going into that in graduate work. But as you referenced, about 10 years ago, a lot of the natural products parts of companies started to be waned off, sort of looking more at structure-activity relationships. Drug companies thought there was a mathematical formula that we could calculate exactly what protein we want to target and make a molecule that looks just like that. As we know, it's not that simple. So natural products are inherently complex and inherently biologically suited. So that's why they're so attractive as drug compounds. And I'm appreciative that you guys have latched onto that as a business opportunity, sort of from the grassroots side. So my question to you guys is, algae is obviously not the only thing that you could pull natural products from in this magnitude. Think about kudzu. There's all sorts of other, whether they're an eyesore or invasive species or things that are in large supply that we may or may not find the value in. Are there other things that you guys are looking at in the future, and is the technology that you've patented for algae transferable for that?
I'll jump right into that. And the answer is, it's always important for any business model to go in an area where you have got an advantage. And our advantage is we can get at biomass that nobody else on the planet can really get at. And it doesn't matter their technologies. They can try it with centrifuges. They can try it with cross-flow filtration. They can do it a dozen different ways. They're not going to come up with a consortia of microorganisms preserved the way we can. So our basic objective is, if it is a single cell or a very microscopic organism that nobody else can get to, that's our interest. We have no interest in pursuing organisms other people can obtain, and that's our major, let's say, filter right now within the space we're looking at. The other thing is, with there potentially being millions upon millions of species, let alone the compounds within these species that have never been studied, never been cultured, that is virtually an unlimited opportunity within the space we're at.
Yeah, John, did you have a question?
Yeah, I've got two questions, actually. One of them sounds kind of silly to
Well,
me,
you'll
but
have to get back in line.
I'll start with that. Yeah. So my silly question, so I keep a boat up in the western basin of Lake Erie, and I pay darn good money to have somebody scrape all that algae off the bottom of the boat each year. Is that, you say you're looking for the things that aren't available, to other people, that other people aren't finding. Is the stuff that's growing on everybody's boats and on the shoreline and everything up there of value? I mean, is that the type of algae that you're using to do your,
It's
your
probably been
testing?
studied.
That's going to be more the macro algae side of it versus the micro. That's a good example of the difference.
So I don't need to start saving all that crud I scrape off the boat.
And don't send it to us either.
I was hoping had some sort of futures there, some sort of profit.
And let me add on to that just for a second, because one of the drug discovery areas that pharma has jumped on relatively recently and had started to find some very interesting compounds is exactly the kind of algae that grows on organisms, in organisms, and is able to be grabbed. We're in the focal area. We're not just grabbing algae. We're grabbing the bacteria, the funguses, everything that grows in that consortia, and we can really look at what those interactions are. So if it can be grabbed, scraped off a boat, pulled off from the bottom of a lake or floating on a lake, and then you can get it with a net, that's not our area of interest.
And then secondly, you talked about cancer research.
We'll come clean your boat.
Might find it interesting, a lot of good slimy stuff. You mentioned cancer research. Does the results that you're, I don't even know how to ask the question, the stuff that you guys are examining, does it show promise in helping with other diseases and other medical applications besides cancer research?
Yeah, and a good example is Lilly. When we first started doing research probably 6 or 7 months ago, before I even joined the company, is that I was primarily working through NCI, National Cancer Institute. Since then, we now are partnering with Lilly, who has, they pretty much participate in probably 65% of the major disease states. And so that's why we're so interested in partnering with them, because we will start to get the data to prove the utility of our biomass across multiple disease states.
Thank you.
Good afternoon. My name is Araya Flynn. I am with the Coalition for Planetary Health and Peace. I'm very excited to be here. Ross knows I have a lot of interest in different things, and plant-based nutrition is one of them. So I'm very excited to see the developments in the algae field, as well as the alternative energy possibilities for it. So I guess I wanted to find out about how long do you think, how far away from expanding algae into fuel are we, and the nutritional possibilities for just consumption.
Terrific. It's a great question. One of the things I would say is there's a number of companies that have been well funded. Solazyme in San Francisco area is a company that actually went public at nearly $1 billion market cap that's focused on nutraceuticals and focused on cosmetics. They're also looking at biofuel applications. So there is an investment community out there that is supporting algae to these other products currently. We get tapped and asked by a number of these different companies for the utilization of our technology to support them. And as recently as yesterday, we had a large multinational startup that was focusing on algae specifically to fuels and the kind of products, nutraceuticals and also foods, that came to us and said, we want to include you in a new FOA or a new answer to a government request to study algae for these purposes. So it's not going to go away. When you talk about the kind of time frame that it'll take, I think that it's everything in time and when there is economic justification for that. It is great to see investments pursuing. People that are aggressive and make investments early, some of them will win big, some of them will lose big. That's just the reality about biofuel type of investments and some of the other products. But we will continue to participate in that. We will continue to focus, and I think that's the objective. So you've got an opportunity, you don't ignore it, but you focus, and our focus is to drive Biosortia Pharmaceuticals to where it can actually contribute and get a return that allows us to do some of these other things we would want to do in another venue at another time.
Thank you.
We have time for just one more, so you're it.
Made it right under the wire. I'm Mark Butterworth. Ross, just wondering, the military's recently announced that they would like to get 10% of their aviation fuel from biofuels. Just wondering if that will increase the long adoption cycle that you just talked about.
It will actually shorten it. So when we look at sponsorships, when we look at infrastructure investment into a new strategy, quite frankly, it's going to cost one way or another. Those dollars will basically be put to use in advancing the technology further than it could be under a highly competitive environment. And it's important, but I think it will shorten the cycle to economic viability. And we are faced essentially with the challenges of how much carbon we put in our atmosphere versus how much carbon we take out of our atmosphere. And algae, the reason it's growing in your lakes, the reason it's growing in your swimming pool, or if you just put water out there in a dish, you will see algae start to grow there, is the fact that those nutrients and the carbon is in the air to support its recycling of those nutrients. So it's going to happen. It's hard to say when. I'd love to say that it could happen within 10 years, but I think I'm a realist, and that is the opportunity right now is to focus on the highest value, because that's where we're going to be able to ultimately create the opportunity to continue down this pathway. You don't want to reach a dead end by doing something that's not economically effective.
Thanks, Ross. Thanks, Kurt. Thanks for some great questions.
Yeah.
Rich, you have the card.
Thank you. For the non-scientists in the group, including myself, I don't think I'll ever look at pond scum or curse the red tide the same way again. It's terrifically interesting, and it's wonderful that you're right here in town doing this stuff. Hope you enjoyed the forum today. You can see it again, as we indicated, on the Ohio channel, or you can find us on YouTube. There's a link to this if you want to take notes from the presentation. There's a link on the CMC website. We can continue the conversation with our guests with coffee and cookies out in the lobby. We remain somewhat green ourselves. The name tags, please recycle. There's a basket out there, drop them in that. If you're interested in any of the Metropolitan Club's forums or whatever else we might be doing, put your card in there, and then we'll come solicit you for membership. We got to hit that $1,000 mark. Don't forget to register for the New at the Zoo, some of which will be here. And let's thank our sponsors once more, Ice Miller and WCBE 90.5 FM. Also, let's thank our speakers, Ross Youngs, Kurt Dieck, and our moderator, Dan Molshako. Thank you all for braving the storm, and we'll see you again soon.